All right, so in just a few seconds, I'm going to teach you how to answer this college exam question. So SDS Page is a lab technique that scientists use to basically separate proteins by size and charge. So we separate different sizes and charges of proteins. And what is this used for? Basically to check for like a purity of a sample of proteins, to see what size of maybe a new protein you're looking at is, or to confirm the identity of a protein. For example, proteins of the same identity would basically yield the same result in this process. Typically use gel electrophoresis for things like DNA and RNA, but this is a process by which we can also do that same process with proteins. So sodium dodecyl sulfate polyacrylamide gel electrophoresis is what SDS Page stands for. Basically, we add a very strong detergent called sodium dodecyl sulfate to a protein. It's going to denature this protein, obviously basically unwinding its 3D structure into basically a singular linear chain of amino acids, which also become negatively charged because the detergent also sticks to them, and so the outside of the protein is going to have this kind of negative charge on it. Then we introduce it into a polyacrylamide gel, which is a gel is commonly used for gel electrophoresis, especially with like smaller molecules. And basically it's a gel, and it's on this plate. You might be already familiar with gel electrophoresis, but basically one side is negatively charged, one side is positive charged. We put it in these wells on this side of the plate, and because it's negatively charged, it's going to want to go this way down the plate, right? But however, just the way polyacrylamide gel is, it has very, very small kind of pores, and so basically larger proteins do not travel as far, as well as less charged proteins don't travel as far. And so yeah, this is SDS Page. Just check out my actual channel for study tips that I found helpful. But yeah, have a good day.